Our next speaker is from UC San Diego, and their talk is the Coccidioides trifactor innate immune detection of fungal conidia, spherules, and endospores by macrophages.
Hi, everyone. My name is Sharon, and today I'm going to be talking about the innate immune detection of fungal Conidia, spherules, and endospores from cocci by macrophages, and specifically looking at how each of the 3 distinct cell forms impact IL-1 beta release and cell death. So previous studies looking at IL-1 beta release have been done on formalin-killed spherules. So I first wanted to look at the impact of dead Conidia of the C. imitis RS strain on bone marrow-derived macrophages cultured in MCSF and primed with GM-CSF. So as a positive control, we can see here that macrophages stimulated with LPS and nigericin have increased cell death and release of IL-1 beta. But when we stimulated cells with dead Conidia, that also led to cell death. However, IL-1 beta release for us was only observed with LPS priming. Interestingly, when we looked at live Conidia, we saw a similar effect. Live Conidia infection led to both cell death and IL-1 beta release, but that only occurred at an MOI of 0.2 and higher. So to determine this type of cell death involved, we treated Conidia-infected macrophages with the pan-caspase inhibitor ZVAD, but that failed to reduce death. And not shown here for the purpose of time, but we also only saw a modest impact of Conidia-infected death on gastomin D, E, MLKL, NELP3, and pyrin-deficient macrophages. And these are all key factors of apoptosis, necroptosis, and other major cell death pathways, which tells us that those cell death pathways are not occurring here. So then we considered ferroptosis, which is an iron-dependent form of regulated cell death characterized by the accumulation of lipid hydroxides, hydroperoxides, and oxidative damage to the cell membrane. And to explore whether ferroptosis might contribute to the death of macrophages here, we use lipoxystatin 1, which is a small molecule inhibitor of ferroptosis. And surprisingly, LIP1 did not block macrophage cell death, but instead increased death. To further explore this, we treated infected macrophages with ferric citrate, which in theory should provide additional iron to fuel ferroptosis. And this treatment led to a reduction in fungal viability and a decreased macrophage death, suggesting to us that macrophage can kill Conidia, and that is rate limited by the access to iron, and in turn, macrophage viability is determined by the level of ferroptotic signaling. To test whether the viability of Conidia in these macrophage cultures is impacted by ferroptosis, we treated the infected macrophages with lipoxystatin 1. And after 24 hours, we see there is increased fungal viability in these macrophages, which further supports the role of lipid peroxidation in Conidia killing. And at the same time, LIP1 did not impact the viability of Conidia alone, confirming that ferroptosis is macrophage-driven. And then to further investigate the inflammasome surrounding ferroptosis, we found that the gastomin D and MLKL deficient macrophages had a lower fungal burden than the wild type here. And that was also the same situation in the lipoxystatin 1 treated cells. And at the same time, we observed increased viability of infected MLKL deficient macrophages, which led us to hypothesize that these knockout macrophages exhibited delayed cell death to increase Conidia killing. And we also saw an increased release of IL-1 beta from the gastomin D deficient macrophages, and hypothesized that gastomin D and MLKL come together to kill Conidia and drive ferroptotic peroxidation. And this is just a little bit further on to the work that we will do. NINJ1, which is another pore former, has been shown to oligomerize and be active downstream of ferroptosis. And so given the modest impact of cell death in these gastomin D and MLKL knockout macrophages, we will be looking at the activity of NINJ1 in these cells. So to start examining the role of spherules in the macrophage inflammasome response, we infected cells with formalin-killed spherules. And again, we saw that they do induce the release of IL-1 beta. And using macrophage deficient in NELP3 and pyrin, which is encoded by the MEF-V gene, we can see that the loss of NELP3 abrogated IL-1 beta release. And then in the pyrin deficient macrophages, there's also a partial defect, which tells us that formalin-killed spherules activate both the NELP and pyrin inflammasomes. So FKC was able to induce macrophage cell death, but this did not require LPS priming, which is very different to what we saw with the dead Conidia. And we also saw that this was dependent on NELP3, caspase 1, and gastromin DE, which indicates to us that formalin-killed spherules induce pyroptosis in these macrophages. And again, this also highlights the very different inflammasome activation from Conidia and spherules. So to see if we can replicate the formalin-killed spherules results with live spherules, we decided to grow them in the presence of macrophages, as Janie has given a beautiful prelude to. And so in the process of doing this, we noted that by lowering the MOI of live Conidia to a 0.01, we were able to get some spherules, but we were able to optimize this process by adding more dead Conidia and using Fungal Finder, which is a custom script that we came up with to account for spherules count and size. We can see that the addition of dead Conidia were able to double the count and the size of these spherules. So with the combination of live and dead Conidia, we were able to generate large spherules up to, oh, it won't play, up to 100 microns. Please come to me if you want to see movies. And simultaneously, we noticed that the addition of the dead Conidia also reduced death induced by live Conidia. So as spherules mature, we were able to notice that by day 3, they began rupturing and releasing endospores, as you can see by the arrows here. So, and they leave behind this ruptured spherule wall. And this post-burst stage is characterized by the release of IL-1 beta, and by FGF, we can also see that the area of these spherules increase. To investigate whether a macrophage can sense these large structures, we utilize a DECTA1 knockout, which is DECTA1 is encoded by this CLEC7A gene. And we saw that they release less IL-1 beta and also produce slightly larger spherules than wild type. And this was really surprising to us that IL-1 beta release wasn't completely inhibited, which suggested that TLR2 may also play a role in recognizing and responding to ruptured spherules. Given the importance of NELP and pyrin inflammasomes in sensing fixed spherules, we hypothesized that it might also be playing a role in this post-burst stage. So we can see from the NELP3 and pyrin single knockout macrophages that IL-1 beta release is inhibited, but you can see by the double knockout that it is completely undetectable. And similarly in the caspase 1E11 double knockout that they're not showing any IL-1 beta release. Our FGF analysis shows that the macrophages forming out of these double knockout macrophages are smaller, and that tells us NELP and pyrin are key players in the inflammasome response. So now looking at the mechanism behind IL-1 beta release, we saw that IL-1 beta release was not affected by individual deletions of gastromin D, E, or MLKL, but the combined deletion of D and E led to reduced IL-1 beta release. And additionally, spherules out of these cultures seem to be larger, suggesting that gastromin D and E might help restrain spherule growth. This one won't play as well. But so since macrophages responded so strongly to both post-burst cultures, we hypothesized that they're responding to these released endospores. And to test this, we infected wild-type macrophages with live endospores isolated from these post-burst cultures. And at an MOI of 1, we can see that they are able to induce a lot of cell death. and they are also able to release IL-1 beta, but this was dependent on NELP3 and not gastromin DE or pyrin. I'll just describe this video. So the endospores go on to release really beautiful spherules, and this was only in our hands shown in the presence of macrophages, which tells us that macrophages is important for cocci to continue on the parasitic cycle under culture conditions. Oh, it's playing. Okay. There you go. I'm sorry about the other video. It's more impressive. To determine if the protective effect of dead Conidia in vitro can be translated to an in vivo model, we infected black 6 mice with 10 to the 2 CFUs of live Conidia, with or without the addition of 10 to the 4 dead Conidia. We saw that mice infected with live Conidia alone started losing significant weight by day 10, and they failed to recover. But mice co-infected with live and dead Conidia maintained stable body weight, similar to our control uninfected mice. And this suggests to us that dead Conidia do provide some sort of protective mechanism against live Conidia-induced weight loss. But we do not think that this protection is due to fungal clearance, because the fungal burden in both lung and spleen looked similar between the 2 treatment groups. And we also looked at the involvement of NELP3 inflammasome in live Conidia infection, and this is with 10 to the 2 CFUs. And so we infected wild type, caspase 111 double knockout, and NELP3 knockout mice. And we saw that the wild type showed they succumbed to infection by day 13, and the caspase 11 and NELP3 knockout mice showed modest resistance. And interestingly, the lung fungal burden in between the genotypes had no difference. And then when we look at the spleen fungal burden, we saw that caspase 111 had a higher fungal load, but not the NELP3. And this suggests to us that perhaps the pyrin is active and compensating for the loss of NELP3 here. So overall, our data shows that cocci infection triggers distinct but overlapping inflammasome pathways in macrophages. And we first show that ferroptosis was important in Conidia killing, and that might be driven by gastromin D and MLKL. Ruptured spherules activate NELP and pyrin inflammasomes, and this is to release IL-1 beta via both gastromin D and E. And the endospores released from these ruptured spherules also go on to activate the NELP3 inflammasome to release more IL-1 beta. And lastly, we show that dead Conidia appears to offer a protective effect in vitro and in vivo. And we also implicated the role of caspase 111 in limiting fungal dissemination, and we'll be looking further into NELP3 and pyrin. So ongoing work, I mentioned NINJ1 earlier. So we're really interested in the role of NINJ1 in the absence of gastromin D and MLKL and how they compensate for the loss of those 2 other pore-forming proteins. And lastly, we'll also be looking at the contribution of TLR2 to Conidia spherule and endospore sensing and IL-1 beta release. And this is our team. Thank you.
So in the face of inflammation, I think of a variety of types, serum iron in humans goes down. Your work is suggesting that giving iron could benefit the host. Is that a true statement?
Yeah.
So I have taught the absolute opposite for years and years and years and years. The model being Yersinia, that clearly if you give iron to people that have ursinosis, they do much worse. I'm standing here terribly impressed with your work, but terribly confused.
Me too. No. So the concept of host immune cells undergoing cell death, whether it be ferroptosis or necroptosis, to sacrifice themselves to limit either fungal or bacterial replication has been shown. And in our hands, it just seems to suggest that iron is important to drive this particular type of cell death. Thank you.